We had people from 27 countries at this conference focused on engineered disease modeling. They're bringing cutting-edge knowledge, breakthroughs, milestones. So this is something that is bringing really a revolution, I would say.
What we've learned about human biology from studying other animals or from studying cells in a dish, it's clear that there's still a lot of gap that remains. That's where engineered human tissue models can help to fill in that gap. It allows us to do testable science that's hard to do in a living animal.
We're working on engineering different types of tissues, from the heart, brain, spinal cord, intestine, to 3D printing of the entire organ. In the future, we would like to regenerate the diseased heart, either with cardiac patches, with blood vessels that are printed, or even replacing the entire heart with a printed heart.
This is the first time we can really study human disease in things that are more complex than just one cell type. We make little versions of organs, and then we try to perturb the system to mimic a disease. And that's a big difference with what we've been doing in the past, in vitro models and animals. The closer we can be to the human biology, the better.
The reason people do animal studies is because regulatory agencies need to ensure safety of drugs that will be tested in humans. Researchers are now working to validate these models so they can be accepted by regulatory agencies, so that you don't need to do testing in an animal. There is another revolution, building automated labs where human mini organs will be grown by robots.
The key takeaway is that while we are able to recreate parts of these organs already, it will need a concerted effort of all the different disciplines working together to really get this translated. This is a very exciting time for bioengineering, and we've seen this field is really developing at speed.